Hello there. So this is our third lecture on immunodeficiency disorders. For this lecture, we will be talking about primary immunodeficiency disorders involving the T cells and some of the primary immunodeficiency disorders that affect both B cells and T cells. For the primary immunodeficiency disorders involving the T cells, we have the following: the George syndrome, Nezeloff syndrome, and class 1 Mhc deficiency. Let's start with the George syndrome. Take note, both the George syndrome and Nezeloff syndrome are characterized by thymic hypoplasia or underdevelopment of the thymus. Now, how will you differentiate a case of the George syndrome from Nezeloff syndrome? Thymic hypoplasia is seen in both the George and Nezeloff syndrome. The underlying problem in the George syndrome is the deletion of the genes in the long arm of chromosome number 22. I have to emphasize that there is that group of genes that can be absent or missing in the case of the George syndrome, but we will no longer cover them in our lecture. The point is, if one of these genes will go missing, then there would be a problem in the development of the third and fourth pharyngeal pouches. And how important are these pharyngeal pouches? They are the embryonic parts that will give rise to the portions of the heart, including the blood vessels in there and the heart valves. And then some parts of the head and neck, particularly the palate, the thymus, and the parathyroid glands. So just try to imagine if one of the genes involved in the George syndrome is missing, then there would be no development or underdevelopment of the third and fourth pharyngeal pouches, and all of these organs mentioned will have problems in their development. Then there would be thymic hypoplasia. And you know very well that the thymus is the maturation site for T cells. So expect that patients with the George syndrome will have problems with producing mature and functional T cells. Pointed by the blue arrow are the third and fourth pharyngeal pouches of the embryo. And then I want you to appreciate from the photo that this is the portion of the embryo that will give rise to the thymus as labeled and the parathyroid glands. What is not shown in there is that the third and the fourth pharyngeal pouches will also give rise to some parts of the heart, the hard palate, and some portions of the head and neck. For you to easily remember the clinical signs and symptoms of the George syndrome, I want you to remember CATCH George. CATCH, letter C, stands for cardiac defects, because you know very well that some portions of the heart will be derived from the third and fourth pharyngeal pouches. Then you have abnormal phasae, which I will explain on the next slide. Then you have thymic hypoplasia, which is the one important for our topic in immunology. You also have the cleft palate. Remember, the third and the fourth pharyngeal pouches will give rise to some parts of the head and neck, and that includes the hard palate. And then there's also hypocalcemia because of the failure of the parathyroid glands to develop. You know very well that parathyroid hormone increases blood calcium level. So if these parathyroid glands will not develop, then expect that the hormone level will be very low, and the consequence will be hypocalcemia. CATCH George. The letter A in the mnemonics CATCH would mean abnormal phasae. Why? Because patients affected with the George syndrome would have specific facial features. Number one, they have small heads. So take a look at the picture of the child on the right side of the screen. Then, they also have low nasal bridge as shown by the blue arrow. And then, these patients would also have epicanthal folds. And then, take a look at the picture of the normal philtrum and upper lip. Patients with the George syndrome will have smooth philtrum and thin upper lip. The left photo is showing you a child with cleft palate, with thin upper lip and low nasal bridge. And then the child on the right photo has a low nasal bridge with smooth philtrum and thin upper lip. And these facial features could be highly suggestive that these children have DiGeorge syndrome. As of this moment, there is no known cure for DiGeorge syndrome. What the doctors can do is to manage bacterial infections with antibiotics, subject the patient to surgery to repair cleft palate and congenital heart abnormalities, and then monitor the blood or serum calcium level and always give the patient calcium supplements because you know very well that the patient has hypoparathyroidism and the consequence of that is what? Hypocalcemia. Aside from calcium supplements, you can also give the patient vitamin D because vitamin D is known to what? Increase blood calcium level. Thymus transplantation can be used to address the absence of thymus, but this is rarely done. Another primary immunodeficiency that involves the T cell is the Neselov syndrome. Again, this one is characterized by thymic hypoplasia. In Neselov syndrome, there is hypoplasia of the thymus, but the patient has normal parathyroid gland function and there is the absence of the other congenital anomalies associated with the George syndrome. So these patients only have one problem, and that is the thymic hypoplasia. The underlying mechanism for Neselov syndrome is still unknown until today. Now, we'll move to Class 1 Mhc Deficiency. Class 1 Mhc Deficiency is an autosomal recessive disorder, meaning you need to get two abnormal genes from your parents before you will manifest the clinical signs and symptoms. And this disorder is characterized by low levels of T cytotoxic or CD8 positive T cells, and I will explain later on why. The underlying reason is the mutation in the TAP gene. TAP stands for transporter associated with antigen processing. This TAP gene encodes for TAP1 or TAP2 proteins. Now, how important are TAP1 and TAP2 proteins? Viral proteins and the proteins produced by cancer cells are degraded by the organelle proteasome. And these proteins are converted into smaller peptides. And these peptides are transported into the rough endoplasmic reticulum. That's the organelle you can find on the 6 o'clock position of the photo. And what will help these peptides go inside the rough endoplasmic reticulum are the TAP1 and TAP2 proteins. Inside the rough endoplasmic reticulum, the peptides are loaded into the class 1 MHC. And together, just follow the arrow, they will be expressed on the surface of the virally infected cell or cancer cell. So that this cancer cell or virally infected cell will have an interaction with CD8 positive T cell or T cytotoxic cell. In the absence of TAP1 and TAP2 proteins, the transport of peptides into the rough endoplasmic reticulum will not be possible. And do you know what will happen? The empty MHC class 1 will be later degraded, thus leading to the failure to express on the surface of the cell class 1 MHC. Thus, the condition is called class 1 Mhc deficiency. Let's go back to this particular slide. According to the last bullet, empty MHC class 1 are degraded intracellularly. Why are they empty? It's because of the failure to transport peptides of viruses and cancer cells into the rough endoplasmic reticulum brought about by the absence of TAP1 and TAP2 proteins. Thus, this one will lead to deficiency of MHC class 1. But the question now is, why is there a decrease in the number of CD8 positive T cells or T cytotoxic cells. And it's also very important to be reviewed that once the immature T cells will reach the thymus, they will be initially referred to as double negative thymocytes. And then later on, these double negative thymocytes will become double positive, meaning each thymocyte or each immature T cell has both CD4 and CD8. And this double positive thymocyte will undergo positive selection in the cortex of the thymus. In positive selection, the cortical cells will use class 1 and class 2 MHC to present antigens to the double positive thymocytes. Those T cells that reacted to the antigen using class 2 will no longer express the CD8. Those T cells that reacted to the antigen using MHC class 1 will no longer express CD4. That's basically the process of positive selection. If the cells will fail to react to the antigens being presented by cortical cells, then these cells are subjected to apoptosis. In MHC class 1 deficiency, the cortical cells will not be able to present antigens using class 1 during the positive selection. And the double positive T cell that is supposed to become the future CD8 positive T cell or T cytotoxic cell will not have any means of reacting to the cortical cell. And the thymus will think that this double positive thymocyte or T cell is defective. And this double positive thymocyte or T cell will be subjected to apoptosis. Thus, none of the double positive thymocytes or T cells will give rise to the CD8 positive T cells or T cytotoxic cells. And that will explain now why people with MHC class 1 deficiency will have low CD8 T cell count. And just try to imagine, they don't have the T cell that is specialized in fighting off viruses. Thus, these patients will have recurrent viral infection and they would develop severe infections with viruses and intracellular bacteria because the antigens of intracellular bacteria are also presented using class 1. At this point, we will be discussing the primary immunodeficiency disorders affecting both the B cells and the T cells. And we have the following: SCID, SCID, or Severe Combined Immunodeficiency Disorders. We have the Wiskott-Aldrich Syndrome, Ataxia telangiectasia, and Reticular Dysgenesis. Let's start with the Severe Combined Immunodeficiency Disorders. Severe Combined Immunodeficiency Disorder, or SCID, SCID, is actually a group of disorders. And these disorders arise from genetic mutations. That's why you have a lot of conditions classified under severe combined immunodeficiency because it can be caused by several genetic mutations. That's why this group of disorder would include the X-linked SCID, the Swiss type SCID, and the rest. The point is, these genetic mutations can cause in the failure of development of T cells and B cells. Another important thing to take note is that the humoral immunity, which is mediated by B cell, can be involved because of two mechanisms. Number one, the disorder affects the development of B cell. So B cell is directly involved in the pathophysiology of the case. Or maybe in the disorder, B cells are produced. It's just that they are not activated because the T cells are non-functional. If T cells are non-functional, it would follow that the B cells will not be activated by the T helper cells to form the plasma cells, and the plasma cells will not be there to produce the antibodies. Take a look at this picture. You have here a hematopoietic stem cell Later on, this hematopoietic stem cell will commit to form the cells of the myeloid lineage and the cells of the lymphoid lineage. For it to commit to form the cells of the lymphoid lineage, this hematopoietic stem cell will be acted upon by interleukin 7. So this cell must have a receptor to interleukin 7. Now, after the effect of interleukin 7, this hematopoietic stem cell will become the common lymphoid progenitor. The action of interleukin 7 on the common lymphoid progenitor will make it differentiate to form the T cells and the B cells. But if that common lymphoid progenitor will be acted upon by interleukin 15, then it will commit to form the natural killer cells. The point is, the hematopoietic stem cell and the common lymphoid progenitors must have receptors to interleukin 7, and for the common lymphoid progenitor, it must have a receptor to interleukin 15. The absence of these receptors will now cause problems with the production of T cells, B cells, and natural killer cells. So since both T cells and B cells are involved, the absence of the receptors will now be classified as one of the severe combined immunodeficiency disorder. Take a look at this receptor for interleukin 7. It has two subunits. You have there the purple colored gamma chain subunit and the green colored alpha chain subunit. In the X-linked severe combined immunodeficiency disorder, there is the absence of the gamma chain subunit. Thus, the hematopoietic stem cells and the common lymphoid progenitors will never respond to interleukin 7, leading to the decrease or no production of T cells and B cells. Interleukin 7 and interleukin 15 share the same gamma chain subunit. Thus, there's also a problem with the response to interleukin 15, and that will affect now the production of the natural killer cells. The first condition under severe combined immunodeficiency disorder is the X-linked SCID. X-linked meaning males will manifest the clinical signs and symptoms while females will not, because females will just act as carriers of the abnormal gene. And females have two X chromosomes. Now, this X-linked SCID is characterized by the mutation in the gene that encodes for the gamma chain subunit of the receptors to interleukin 7 and interleukin 15. You know very well that the hematopoietic stem cells will only commit to form the common lymphoid progenitor in the action of interleukin 7. The absence of the receptor to interleukin 7 will impair the ability of the hematopoietic stem cells to give rise to the cells of the lymphoid lineage. And another important function of interleukin 7 is to enhance the differentiation of the common lymphoid progenitor to form the T cells in the B cells. Again, interleukin 7 receptor has alpha subunit and gamma chain subunit. And this gamma chain subunit is absent in the X-linked severe combined immunodeficiency disorder. Before I can make you understand the Swiss type severe combined immunodeficiency disorder, I have to review you on the basics about nucleic acids, particularly DNA. So take a look at the picture of the double-stranded DNA, and then appreciate that each of the strand is composed of series of nucleotides. Shown on the right side of the screen is one nucleotide, and it has three basic components: the yellow-colored phosphate group, and then the purple-colored sugar group, and the green-colored nitrogenous base. The components of the nucleotides are the following. We have the green-colored sugar. Attached to the sixth carbon is the phosphate group. Attached to its first carbon is the nitrogenous base. This nitrogenous base could either be a purine or pyrimidine. A purine is easy to identify because of the presence of two fused rings. And there are two types of purines in the DNA. You have adenine and guanine. In the case of pyrimidine, we have thymidine and cytosine in DNA, and we have cytosine and uracil in RNA. When cells in the body die, they release their nucleic acids. One of the metabolic byproducts of DNA is deoxyadenosine. So for you to picture out how does this one look like, just think of the deoxyribose sugar, and attached to its first carbon is the purine adenine. This deoxyadenosine is toxic to developing T cells and B cells. Thus, it should be eliminated by the body. How is it eliminated? With the action of adenosine deaminase enzyme. And this deoxyadenosine is converted to deoxyinosine. Now, in people with Swiss type SCID or severe combined immunodeficiency, they have this mutation in the gene that encodes for the enzyme adenosine deaminase. Thus, they cannot convert deoxyadenosine to deoxyinosine, and there will be a build-up in the body, particularly in the blood, of deoxyadenosine. So just try to imagine, this one is toxic to developing T cells and B cells. Aside from that, that deoxyadenosine is converted to deoxyadenosine triphosphate, which is also toxic to developing T cells and B cells. Thus, Swiss type is now classified under severe combined immunodeficiency disorder because this particular group of disorder is classified under primary immunodeficiency disorders affecting both B cells and T cells. Swiss type SCID is an autosomal recessive disorder, meaning you need to get two abnormal genes from your parents before you will manifest the clinical signs and symptoms. And this disorder is characterized by the absence of the adenosine deaminase enzyme, leading to the accumulation of deoxyadenosine and deoxyadenosine triphosphate, which are very toxic to immature lymphocytes, leading to their depletion. Thus, there is the involvement of both T cells and B cells in the process. The next type of SCID is the Bare lymphocyte syndrome. This is characterized by the absence or mutation of the genes that encode for the transcription factors that allow the cells, particularly the monocytes, the macrophages, and the B cells, to express MHC class 2. Now, if there is no MHC class 2, then there could be a possible consequence. What is that? We will discuss that in a while. Take a look at the picture of MHC class 2. It has two subunits. You have the alpha and the beta. Both of these subunits are proteins by nature. So meaning they must have corresponding genes that code for them in our DNA. And for these alpha and beta subunits to be produced, you need to have two important processes: transcription of the gene and the translation of the messenger RNA. You have here a double-stranded DNA, and DNA is known to contain the genes, and genes encode for proteins. So let's say, for example, the RNA polymerase there has separated the two strands of DNA because in one of the strands you have there the genes that encode for the alpha and beta subunits of the MHC class 2. The thing is, that RNA polymerase should copy complementarily the bases in the gene into messenger RNA. But that RNA polymerase cannot do that without the help of transcription factors. In the Bare lymphocyte syndrome, there is the absence of the transcription factors that will allow the transcription of the genes that encode for the alpha and beta subunits of MHC class 2. So the cells in the body that are supposed to express class 2 cannot express them. Let's have a short review. You know very well that the genes that encode for class 1 and class 2 MHC are found in the chromosome number 6. Take a look at the picture. You have here red color antigen presenting cell and is supposed to activate the gold color T helper cell, but the antigen presenting cell cannot express the MHC class 2 because of the loss of the transcription factors. Thus, these antigen presenting cells cannot activate the T helper cell. So the T helper cell in this case becomes non-functional. So if the T helper will become non-functional, expect that the B cells will no longer be activated to form the plasma cells, right? So the absence of MHC class 2 will result to non-functional T cells and B cells. So this is really a form of combined immunodeficiency. The absence of MHC class 2 in bare lymphocytes will also have an effect on positive selection. So the cortical cells will not be able to present antigens using MHC class 2. And what will happen? The double positive thymocyte or T cell that is supposed to give rise to CD4 positive T cells will not be able to react to the cortical cells. Thus, these double positive T cells will be subjected to apoptosis, leading to decreased CD4 positive T cell count. And if these T cells are of low levels, then expect that the functions of the B cells will also be greatly affected. Another form of severe combined immunodeficiency disorder is the absence or deficiency of the purine nucleoside phosphorylase enzyme. The main reason behind this one is the mutation in the gene PNP. PNP gene encodes for the purine nucleoside phosphorylase enzyme. Now, what will be the consequence if this enzyme will go missing? DNA metabolites will no longer be eliminated, especially those metabolites that are acted upon by purine nucleoside phosphorylase enzyme. Now, these DNA metabolites are very toxic to immature lymphoid cells, causing them to die. And these patients will have low lymphocyte counts or lymphopenia. And that's the very reason why this one is classified under severe combined immunodeficiency, because both T cells and B cells are affected. Take a look at these biochemical reactions. The DNA metabolites that are eliminated by the enzyme purine nucleoside phosphorylase are inosine and guanosine. Please recall that inosine is derived from the adenosine, which is a part of the nucleotides of DNA. And this adenosine is converted to inosine by the enzyme adenosine deaminase, which is absent in Swiss type SCID, right? One of the portions of the nucleotides is also the guanosine. And this guanosine is eliminated by purine nucleoside phosphorylase enzyme. Now, what will happen is inosine is converted by PNP enzyme into hypoxanthine. Hypoxanthine is converted to xanthine, and xanthine is later metabolized to form uric acid. And this uric acid is eliminated by the kidney through the urine. So that's how we get rid of the DNA metabolites, especially those that come from the adenosine. You have adenosine being converted to inosine, and then to hypoxanthine, then to xanthine. In the case of guanosine, it is converted by PNP enzyme to guanine, and then later on to xanthine, and that xanthine will be converted to uric acid. So just try to imagine, if the person is born with mutation in the PNP gene, inosine and guanosine are not eliminated from the body, and these two are highly toxic to immature lymphoid cells, causing them to die or decrease in number. Now, what are the possible treatment options for individuals with SCID? You have aggressive antibiotic therapy. Aggressive because you will be dealing with a lot of organisms because you have impaired T and B cell functions. And then you can also go for hematopoietic stem cell transplant. And if this will be allowed in the future, then gene therapy can also be an option. We have already finished discussing the group of diseases that fall under the severe combined immunodeficiency disorders. Let's try to recall. We have the X-linked, we have the Swiss type, we have the Bear lymphocyte syndrome, and we also have the Purine nucleoside phosphorylase enzyme deficiency. Now, we'll go to Wiskott-Aldrich syndrome. Wiskott-Aldrich syndrome is another X-linked disorder similar to chronic granulomatous disease. You have the Bruton's X-linked agammaglobulinemia and the X-linked SCID. This particular disorder is characterized by a clinical triad, and I want you to memorize them by heart because most likely they will come out in your board exam review materials and even in the board exam. So you have thrombocytopenia or low platelet count. We have eczema and recurrent infections, which could mean that there is what? Immunodeficiency or immunocompromised state. Now, the problem in this particular disorder is the mutation in the WAS protein gene. WAS protein gene stands for Wiskott-Aldrich syndrome protein gene. And this gene is particularly found on the short arm of X chromosome. In this picture, you have the antigen-presenting cell and the T cell. On the surface of the antigen-presenting cell, you have there the green-colored MHC class II with the bacterial protein loaded onto it. Beside it is the purple B7. I hope you can remember that B7 is a co-stimulatory molecule during antigen presentation. On the surface of the T cell, you have there the T cell receptor. That's the colored blue structure displayed on the surface of the T cell. Beside it is the violet CD28. And then, take a look at the cytoplasm of the T cell. You have there a structure that would stabilize both the T cell receptor that contains the CD3 and the CD28. And that structure is F-actin. F-actin is one of the cytoskeletal frameworks of the cell. I hope you can remember the terms intermediate filament, microfilament, microtubules. That is where the F-actin is categorized under. Now, the point is, this F-actin is organized with the help of the Wiskott-Aldrich syndrome protein. In individuals with Wiskott-Aldrich syndrome, there is a mutation in the Wiskott-Aldrich syndrome protein gene, which will now cause the absence of Wiskott-Aldrich syndrome protein. In the absence of such protein, then that F-actin will no longer be formed. And the T cell receptor with the CD3 and the CD28 will not be stabilized, thus affecting the activation of the T helper by the antigen-presenting cell. Now, if this T helper is not activated, it would follow that this T helper will not activate the B cell. Thus, there is what? An involvement of both T cells and B cells. Wiskott-Aldrich syndrome protein is expressed exclusively by hematopoietic stem cells, and it's very important in the formation of the cytoskeleton, particularly the actin. Its absence would have an impact on the immunologic synapse, and that is where the T cell and the antigen-presenting cells are interacting with each other during the antigen presentation. Now, in individuals with this condition, their T cells have problems with organizing the actin cytoskeleton in their cytoplasm, leading to poor stability of their T cell receptor with CD3 and CD28. Thus, they will have a problem with adhering and interacting with the antigen-presenting cells. The B cell function is affected because there is the failure to activate the T cell. And take note of this, for an unknown reason, the T cells and B cells of individuals with Wiskott-Aldrich syndrome are short-lived. Thus, this is really an example of a primary immunodeficiency disorder that is affecting or involving both B cells and T cells. The cytoskeleton of the cell has something to do with its shape. In the case of platelets, since there is the absence of Wiskott-Aldrich protein, which is important for the assembly of the cytoskeleton, these platelets will have problems with maintaining their normal shape. Thus, these platelets will be sequestered and trapped in the spleen, leading to their destruction. Thus, patients with Wiskott-Aldrich syndrome will have thrombocytopenia. And they will have what? Oral mucosal bleeding or gingival bleeding. You have petechial rashes on the skin, epistaxis, the bleeding in the nose, and worst, they will also have internal bleeding, especially on the gastrointestinal tract. Aside from the problem in the shape, platelets of patients with Wiskott-Aldrich syndrome tend to be smaller than the normal platelets. So I want you to take a look at the normal sized platelet in this blood smear and compare that to the small platelet that is seen in cases of Wiskott-Aldrich syndrome protein. So with this size, do you think it would be easy for the platelets to form the platelet plug and stop the bleeding? It's not, right? That's why people with Wiskott-Aldrich syndrome would have bleeding disorders. Let's try to compare these two blood smears. Blood smear A was taken from a normal individual, and then take a look at the size of his or her platelets. And then compare these platelets to the platelets of blood smear B. Blood smear B has smaller platelets because blood smear B was sampled from a patient with Wiskott-Aldrich syndrome. This child has Wiskott-Aldrich syndrome. And then take a look at the petechial rashes on his skin. Those rashes would result from thrombocytopenia or impaired platelet function. The clinical triad for Wiskott-Aldrich syndrome includes immunodeficiency or recurrent infection, thrombocytopenia, and eczema. This patient in the photo has Wiskott-Aldrich syndrome, and this patient is having atopic dermatitis like eczema. The main reason behind the eczema in Wiskott-Aldrich syndrome is still unknown until today. So how do we treat or manage patients with Wiskott-Aldrich syndrome? We can go for hematopoietic stem cell transplantation, which is nice because it will correct the thrombocytopenia and the immunodeficiency. If we will identify infections, then we should give the patients antibiotics. If the patient is having bleeding episodes, then we can do platelet transfusion or give the patient thrombopoietin. This thrombopoietin is a hormone produced by the liver that will make the hematopoietic stem cells in the bone marrow commit to form the megakaryocytes, which are the precursors of our platelets. And since these patients are having thrombocytopenia and around 70% of the platelets in the body are sequestered or stored in the spleen, removal of the spleen or splenectomy will somehow help in the correction of the low platelet count because the platelets will no longer be stored in the spleen. And since there is impaired B cell function, we can also give the patient intravenous immunoglobulins. And that ends the third lecture on immunodeficiency disorders. In the next video, we will be talking about the remaining primary immunodeficiency disorders affecting both B cells and T cells, those that involve the natural killer cells, and we'll have a short review of the immunodeficiency disorders affecting the proteins of the complement system. Thank you very much for listening.